Now moving to the microbiology section. Firstly entered, we see 3-foot corridor. After that we enter to the changing room, where we remove the outside shoes and lab coat and wear the sterilized gown or lab coat. After that we cross this table safely and move to the airlock room 1. This is the airlock room 1. I have a filter. After that we move to the airlock room 2. Then we enter to the 4-foot corridor. Here we have 4 sections. Firstly we have sterility section. This is the sterility room, where we sterilize the normal water, saline water, apart from sterilized product under the biosafety cabinet. Now moving to the second section. That is Pharma Quality section. This is the Pharma Quality section, where we analyze the pharmaceutical products, that is capsule, tablets and ointments under the laminar airflow. Now moving to the third section, that is culture handling section, where we have biosafety cabinet. Under the biosafety cabinet we analyze and testing the food samples, culture maintenance, and also biochemical and pathogen testing. We also have separate refrigerator and water bath for culture maintenance. Now moving for the last section, that is food MLT section. This is the laminar airflow, where we test the food samples. After that, this is the dynamic pass box, which is open one time from one side only. After that we have trolley, and now move to the exit section, that is airlock 3. And then airlock 4, where we discard the gown and remove the lab coat. After that we are again come to the 3-foot corridor, and now moving to the media instrument section. Here firstly I introduce the autoclave, that is used for steam heat sterilization. Then moving to the BOD incubator, which is set in the 35 degrees Celsius temperature. After that we have hot air oven, which is used for dry heat sterilization. As you know, we have 3 different incubators, which is set into the 3 different temperatures. And this is the second BOD incubator, which is set in the temperature of 25 degrees Celsius for the growth of yeast and mold. This is the dynamic pass box. This is the water bath. This is the weighing machine, where we made the media. This is the digital colony counter, where we count the colonies. Calculator. It is the pH meter. This is the binocular microscope. This is the dry bath incubator, and last but not the least, it is the UV light. That we move to the washing area. That's all. Thank you.